Okay, so now I'm taking a look at the extra microRNA problems that we talked about in class. These were dealing with miR200B and miR429 in mice and their effect on fertility. And this was based on the reference Hazuwa et al. Okay, so and this question is on page 436 of our textbook, or these questions. And again, you can find the reference on ReggieNet in Lecture 20 folder. So, first question. Based on the results described above, draw a diagram to explain possible relationships between miR220B and 429, the ZEB1 gene and the LHB gene, and ovulation in the miR DKO female mice. Okay. So again, you would have to be to understand this, you would kind of need to read those lecture slides and try to answer the question on your own. But I'll answer it here as well. And if you're already thinking about it, then you might understand what I'm about to go over. But if you're not thinking about it, please read the slides 14 and 15 from our PowerPoint and try to answer these questions on your own first. So what is going on here? It looks like miR200B and miR429 make microRNAs, or are microRNAs, that are used by the RISC complex. And what they do is that they suppress a gene called ZEB1, and I think I was calling this ZERB1 or something in class, but we're now that I can see it in the book right here, it's ZEB1, and ZEB1 is a transcriptional repressor of the gene called LHB. LHB is the gene for luteinizing hormone, LH. LH you need, or females need, to release oocytes during ovulation. So oocyte release during ovulation. Okay, so what appears to happen in female mice is that in the ovaries, or in the appropriate cells, or I'm sorry, wherever luteinizing hormone is made, by those cells, wherever those cells are, I think it might be in the pituitary gland, but that's not important. Wherever luteinizing hormone is made, so the miR200B and miR429 genes are expressed. These are expressed. These microRNAs suppress translation of the ZEB1 messenger RNA. The ZEB1 messenger RNA, the ZEB1 protein levels go down. As a result, it cannot repress the LHB gene. As a result of failure to repress LHB, luteinizing hormone goes up, and because luteinizing hormone goes up, the ovaries go through ovulation and release the oocytes. Okay, so that's the mechanism, and that's what we can learn from this study. And because humans conserve all those genes, then it probably works the same way in humans. But so what happens in the double knockout mice, right? Okay, so in the double knockout mice, so in the microRNA, the miR200B, miR429 knockout mice. So we don't make those mice don't make those microRNAs. As a result, the ZRB messenger RNA is translated, and we got lots of, I said ZRB again, didn't I? ZEB1 gene can be translated. We have lots of ZEB1 protein. As a result, LHB is suppressed. So we don't express enough LHB. We don't release enough luteinizing hormone, and we don't ovulate, leading to female mice that are mostly infertile or have significant reductions in fertility. So I thought this was a great example, 2013, very recent paper, of how scientists are trying to figure out how these microRNAs regulate genes in mice and and also humans, by using mice as a model to understand microRNA-based regulation of gene expression. So the next question is, in this study, microRNA double knockout female mice were mated with male mice overexpressing transgenes for microRNA 200B and microRNA 429. So the males are making lots of this microRNA. So males overexpressing OE, that means making lots of microRNA 200B and microRNA 429. So would you expect the pregnancy rate of the miR-DKO females would increase, decrease, or stay the same as the microRNA DKO female mice crossed with heterozygous males? And they crossed them, in the study, they crossed them with heterozygous males for some reason, to show that the mice were mostly infertile. The female mice were mostly infertile. And I don't think it mattered that they used heterozygous males. They could have used wild-type males too. As far as I know, I don't think it mattered. But what we're wondering now is what would happen to the pregnancy rate now that we're crossing overexpression males that are making tons of these microRNAs, to females that are double knockouts for both those microRNAs. So we mate these together, and what happens? Do the females still look infertile? Or do they show normal fertility now that we're crossing with these males over here? Or do they look super fertile? You know, do they make lots of little mouse babies? So we are going to make a guess there, and I'm going to say they're going to be the same. I think they're going to be the same, because who cares what the microRNAs are doing in the males? We already know when the males are deleted of both those genes that they show no change in fertility. Why would making more make the males super fertile? And why would making more of those microRNAs in the males have an effect on how the microRNAs are regulating genes in the females? So, you know, the males aren't transferring those microRNAs to the bloodstream or to the cells of the females that need to suppress ZEB1 in order to undergo ovulation. So I'm going to say fertility rate is going to remain the same, which that means the female double knockouts are still going to be mostly infertile or show reduced fertility relative to wild-type females. So I've taken a look at those, at the reference and the supplementary materials. Sometimes these papers will have a couple pages where they describe the main results in the discussion, and they'll have like 20 or more pages of supplementary material. And as far as I can tell, is this question is completely inaccurate. So it seems like I think the authors of this textbook made a mistake, and I don't think that they did this particular experiment in this paper in the reference. So I think the authors of our book misinterpreted what was going on there. So we are just going to have to move on to the next part of this question and just assume that we're probably right here by guessing that overexpressing those microRNAs in the male likely didn't have an effect on overall fertility. It seems like they did something like that in the experiment, but there wasn't any data showing fertility levels in the female DKOs from this particular cross. Okay, so let's take a look at the last part of this problem. So using OMIM, and let's figure out what that is. So OMIM is the Online Mendelian Inheritance of Man website. We're going to use that to identify the human homolog for ZEB1 and find out if there are any human disorders associated with problems with this gene. And the final question is, do these surprise you? So let's see if I can actually, instead of working with paint here, maybe I can actually look at, I can do a screencast of my Chrome window. Okay, so let me switch to that. Okay, so I have the Online Mendelian Inheritance in Man web portal open up, and our question wanted us to look for the homolog of ZEB1. So this is just the homolog. We talked about orthologs a little bit. So homologs is a generic term for a bunch of different logs, like ortholog and paralog, and so we're going to be looking for an ortholog, but we can use the term homolog here. That's fine too. So let's see if we can find the human homolog for ZEB1, and let's just type ZEB1 and see how easy it is. We've got a lot of different ZEB1 genes here, so looking good so far. Okay, so here we go. Zinc finger box binding homeodomain ZEB1. So I'm going to click on that, and lots of different things here. So HGNC approved, gene symbol ZEB1. So this is should be a human specific website. I actually don't work with humans, so or don't do research on human genes because I work in fungi. So I don't ever. This is my first time looking at this site, but it's pretty cool. And so we have a description of ZEB1. ZEB1 encodes a zinc finger transcription factor that represses T lymphocyte-specific interleukin-2 gene expression by binding to a negative regulatory domain 100 nucleotides 5' of the interleukin-2 transcription start site. So we've looked at ZEB1's transcriptional repression of luteinizing hormone, but apparently it expresses suppresses a bunch of different things. So suppressing luteinizing hormone might just be one function, and here we're seeing it also suppresses interleukin-2, which is a gene involved in the immune system. Okay, let's see here. Let's see if we can find luteinizing hormone. And here, okay, here we have the reference. So Hasawa described anovulation and infertility in female mice lacking the microRNAs MRI-200B and MRI-429. Both microRNAs are strongly expressed in the pituitary gland, where they suppress expression of the transcriptional repressor ZEB1. Eliminating these microRNAs in turn inhibits luteinizing hormone synthesis by repressing transcription of its beta subunit gene. Which leads to lowered serum LH concentration and impaired LH surge and failure to ovulate. Asawa et al. Concluded that these results revealed roles for miR-200B and miR-429 and their target ZEB1 gene in the regulation of mammalian reproduction. Thus, the hypothalamal pituitary ovarian axis requires miR-200B and miR-429 to support ovulation. Wow, okay, that is pretty amazing. This is a pretty amazing website, and I think it's relatively easy to understand. And if you can understand this as a undergraduate genetics student, that's amazing. There's a lot of information here we can get on ZEB1. And so the question was, using OMIM, identify the human homolog for ZEB1 and find out if there are any human disorders associated with problems with this gene. Okay, so let's see, are there any disorders? Molecular genetics. So I wonder if there's specific, they look like there's a bunch of things here. So let's see, allelic variants, 4 selected examples. So my take from this site is that we learned some things about human ZEB1. For instance, mutations in ZEB1 have led to diseases called, a lot of eye diseases here, so corneal dystrophy, a couple different cases, posterior polymorphous corneal dystrophy, and Fuchs endothelial corneal dystrophy. So am I surprised that mutation in these would lead to these diseases from the study that we just talked about? So I would say, well, yeah, I guess I'm kind of surprised, right? Because this study was mostly about the effects of ZEB1 on suppressing luteinizing hormone, right? So ZEB1 suppresses luteinizing hormone. So I guess I was expecting for, well, I don't know a whole lot about luteinizing hormone, but I was expecting to see some fertility defects, but we are seeing defects associated with the eye for some reason. So I would say that this is surprising to me. On one level it is surprising. On another point is I might be missing something, but it's also possible that, you know, ZEB1 is just an important gene, and when you mutate it, well, then a lot of things go wrong, and the one that seems to be at least the easiest to detect is problems with eyesight. So not the best problems here, but I like the study, and I like this study for introducing how microRNAs work. I think our textbook could have done a little better job on asking us to explore these other important things like the OMIM pathway, and actually looking at a reference. I think they could have done a better job of asking questions that had better or more obvious answers. But anyways, I hope you enjoyed this problem, and it's kind of the first time we've looked at a real reference and used the internet to look at a human gene. So okay, I hope you liked it. So there won't be any questions on the exam based on questions 2 and 3 on slide 16, but you should understand how it seems that these microRNAs regulate ZEB1 and LHB and ovulation in mice.